Good evening. My name is Johanna Koljonen and you are watching Crosstalks, a collaboration between two of Sweden's leading universities, KTH, the Royal Institute of Technology, and Stockholm University. This is the 24th episode, and just like always, we'll hear from some world-leading scientists as well as the students in our audience. And don't forget, our Twitter handle is @CrosstalksTV. You can also use our hashtag Crosstalks. Tonight, we'll delve deep into inner space. Literally, millennia of work on medical science has given us the ability to cure infections, transplant organs, and fix broken bones. But zoom in close enough on the human body, and you'll find that many mysteries remain. Some of today's most interesting research into human physiology is done on a true micro level, and understanding these, our smallest building blocks, is essential for the diagnosis and treatment of common diseases and could also provide new insights into aging and other processes in the body. With us today in the studio are three researchers from SciLifelab, which is a collaboration between four of the country's largest universities. Mathias Uhlén is professor of microbiology at the KTH Royal Institute of Technology, program director for the Human Protein Resource Project, and director of SciLifelab. Nilsson, professor at the Department of Biochemistry and Biophysics at Stockholm University, and site director for SciLifelab in Stockholm. And Erik, Lindahl, professor of biophysics at Stockholm University and at KTH, the Royal Institute of Technology. Please give them a warm hand. Mathias Uhlén, you're currently working with the Human Protein Resource Project, which is working to map the human proteome. What's the most interesting thing about this project, in your opinion?
Well, first of all, I think that you stated rather nicely, we still have some mysteries when it comes to our own body. I mean, what we're trying to do in this project that we started about 10 years ago now is try to map all the proteins, all the building blocks of human beings. And we know now that you and I and everyone else here has about 20,000 building blocks, Lego pieces, in the body. And they are there to make us Kalle or Pelle or whatever human being we are. So what we try to do in this project, which is led from here from Stockholm, is to go through one by one and then say, where are they in the body? We're also trying to say what they are doing and so on. And to in the end, have a map, an atlas of all the building blocks of humans. And obviously this is also interesting from the point of view that all the drugs that we have and are targeting these proteins. So obviously to know the map is also how to then create new drugs and new treatments.
What do you hope it will lead to? Is it a medical science question primarily? Is it about solving diseases?
I guess this is very much basic research in the first instance. So we will, in the end of 2014, we will release the first complete atlas of the human proteome and the human protein.
In just a few months?
Yes. And we have spent about 1,000 man-years to do this. We have spent about 100 IT hours of IT years on software development. We have about 13 million images to try to explain how we look like in the body. And I think this can be used to understand human beings, which I think is very important from a basic point of view, but also then to develop new drugs.
I almost ask an amateur question, which is when we have this map, can we build humans? I realized, of course, we are building humans all the time. We don't need the map for that particular
No,
thing.
I think it's going to be very hard to build humans, I think. We are
from the pieces, yes.
Yeah, we are very good at doing that ourselves, but that's through the normal way, I think.
Yes.
That's
a relief, at least.
Yes.
Mats Nilsson, your research is in molecular analysis. What's the most interesting frontier in your field of research right now?
So what I think what we've seen is a dramatic development of technologies to measure the building blocks. So the kind of work Mathias is doing, but also in other fields, not least in the DNA sequencing field, we now can sequence whatever we like. We can measure the molecular composition of any organism, any cell in the body. And that is sort of the the construction map for the proteins of all organisms. So this development of tools to measure molecules makes it now really possible to do the things that Mathias is describing, and in all classes of the building blocks, the DNA and RNA and the proteins. So that makes I think the next level is to see what is the molecular inventory of all cells in the body, to try to understand the modules, the functional modules in cells, how have a molecular definition of all cells to better understand how they interplay in organs and execute the functions, the physiological function in the body. So take it from the molecules to understand how they connect in cells, and see how cells connect into tissues and the functions of any living organism.
Erik, Lindahl, you have a slightly different background than the other two gentlemen. You come from physics and mathematics, and your specialty is combining bioinformatics and life science. Could you tell us a bit about what that means and what your current research interests are?
Actually, it's fun when you mention that, because I occasionally forget that I'm not a chemist, a real chemist like Mathias and Mats. Although I think the difference is that chemistry has historically very much been a top-down approach, and coming from physics, the original idea there is that all these reactions you see, and remember, even the creation of babies or something is ultimately a chemical reaction. Those all obey the laws of physics, right? We can understand why a protein falls or something from physics. The problem is that historically we have not been able to do it. It's a really, really complicated problem. It requires immense amounts of computing power. But from our point of view, one of the most amazing developments the last couple of decades is that we now have access to this computing power. We have amazing computer resources here in Stockholm. And what this is gradually enabling us to do is that I can't do the things like, say, Mathias can in the Human Protein Atlas. But we can, say, looking at 1, 2, or maybe 10 proteins, we can actually predict in the computer what will happen if I replace one of these small Lego pieces, or why does a particular drug, say alcohol or an anesthetic or something, have the action it has in your brain. And the fascinating thing that's gradually happening there now is that we're moving from, say, 10 to 100 or 1,000 molecules, and suddenly we can start doing many of the things in the computer that Mathias or Mats normally do in the lab.
I'm going to have to ask a layman's question again. Are those models reliable? A sort of a layman's worry would be that the more, for instance, the more molecules you add into your analysis, the more proteins you're working with, and simultaneously, the more complex the relations between them become. Are the mathematical models sound?
Now, that's a really good question. From one point of view, you could argue they're not, but it doesn't necessarily matter. Because the trick here is that had I worked with one molecule, I would be really worried, right? But if I see the same thing for 1,000 molecules, that's essentially the same approach we're using in the lab. Now, a sequencing machine could be wrong one time out of a million. But the point is that we're no longer relying on these individual results. I would say that a unifying factor by the things that all three of us do is that we're working with high throughput data. And surprisingly enough, that of course means the data itself is very important. But since we're focusing on the statistics of the data, that also means that we are getting better and better at handling the errors. There are errors in the data, but we have to learn to live with it.
I'm realizing from what you're saying that, which of course I should have understood from the research already, that this is now a field where so many different disciplines have to collaborate. I think if you're a grad student today and looking at a path to choose, you could get excited about building the tools that will help you do these things, or whether it's about physically analyzing molecules, or whether it's mathematically modeling them, and then it's also about doing the work, doing the analysis work, or being the chemist on the project, so on, or being the engineer who builds the tools. And I am realizing as I'm saying this out loud that maybe indeed this is what the SciLifelab is all about. So Mathias, you are currently the head of SciLifelab, and this is a joint initiative between 4 universities. Can you just tell us just very briefly about the mission statement of the SciLifelab?
But I think you just said it very nicely, is that what we're trying to create at SciLifelab or Science for Life Laboratory is to try to create an environment where a lot of disciplines come together. We want to have the medical researchers, the clinical researchers in the same building as we have natural science researchers, physicists, but also then experimentalists from different disciplines and actually also different faculties. And we're trying then to create an environment where we can go sort of technology-driven, data-driven, but use that to study both medicine but also environment. And we spend a lot of time trying to put together these kind of groups that can then tackle new problems, issues, but also ideas. So for example then, we have very, very powerful ways to analyze DNA now. About 10 years ago, it took about 5,000 people, 10 years and $2 billion to sequence one DNA from one human being. Now we can do it, we can do about 20 such analysis per day, and it cost about $1,000. So it's, and it costed $2 billion just 10 years ago. So it's an incredible development that we're seeing. But it also creates problems. And one of them is just like Erik is saying, is that we now produce so much data that in order to go from data to knowledge, we need to sort of be there and have people like Erik and others that can actually take us from data to knowledge. And then of course what we hope in the end is that we can go from knowledge to products that will help mankind. And that will of course take us even longer. But I mean, we're still doing quite well, I think.
Given what's happened in 10 years, though, we have to maybe start to question the idea of what's a long time in science now.
Yeah.
Maybe a long time is 4 years.
Yeah, but I think it is amazing what's going on, and to only think about that we would know all the building blocks of human beings only 10 years ago was almost hard to imagine. And here we are now with complete maps and so on.
Well, I realize,
but still,
yeah.
I should say that I still think that we will have to spend a few hundred years to understand human beings. So I still think it's a very good idea if you're thinking about research that you should go into life science, because I think it is so complex. There are few building blocks, but they interact in such a magical way that it's hard for us to understand it, and so it's still a very good field to move into, I think.
Mats, you've been involved in, among many other things, in developing a new technique for studying cell tissue without the need to break down the tissue completely. Could you explain a little bit about what you're hoping to find out?
Right. So what we have recently developed is a technology to enable sequencing in cells in intact tissue. So without having to extract the nucleic acid from the tissue and then determine.
Could that be on a living person?
No, it has. Well, no, it has to be in fixed tissue. So
okay.
it's from biopsies or material that have been removed from people, or it's from animals. So with that we can have again make maps in a much more detailed way than before, where we can study mRNA expression in tissues and see how the mRNA molecules distribute across the tissue in much more comprehensive way than it was possible to do just until recently. I would like to maybe also connect a little bit with this cross-disciplinary thought. It has been molecular biology was really founded by physicists in the 30s or so that really wanted to be able to compute biology, and for the benefit of medicine. That was sort of the idea of trying to understand biology, not only describing biology, but try to understand and compute. And I think now, so I would say molecular biology is beginning now, because now we have the molecules, we have the tools to measure the molecules that are necessary to try to start to compute biology. So this is really the starting point. So I agree completely with Mathias. This is it's not like we can close this field. It's just beginning.
Erik, you've also been one of the initiators for new education program in bioscience at SciLifelab. What's special about that?
I think it mostly comes out of our own needs and the things that we've described here. And I like the way Mathias stressed how fast this field has developed. And I just thought of this analogy that 7 years ago, the first smartphones came along, and that feels like an eternity, right? What's important to realize, that development is nothing compared to what's happened in this field. This is so much faster. And of course, we're in the situation now that 8 years ago, there were no app developers. And the problem, we don't have the people, because the people who are going to work in this, they need to understand computers, they need to understand medicine, they need to have a very deep understanding about these techniques, how we're going to develop the new technology. And there simply aren't any education programs out there today. And we can't really expect people to have a degree both in engineering and natural science and medicine, because they would be 45 before they started, right? So the 3 universities in Stockholm...
but we don't have. But by then everything will have changed again, right?
Exactly. Right. So our 2 universities, together with Karolinska Institute here in Stockholm, have joined forces, and starting next fall, so fall 2015, the first students in the new joint master's program in molecular techniques and life science are going to start partly at SciLifelab. Which is quite exciting. So it's the first 3-way master's program, at least in Sweden and possibly in Europe.
Do they have to study 3 times as long, though?
No, they don't. And I think this is one of the challenges for us, because, of course, at some point you need to condense this down to 2 years. And what is it that we should cut away? And there are, of course, many things that you do have to cut away, but I think the most exciting things is that you're bringing up a new generation of students who, from the very first day, they're just as used to standing in the lab as you are in hacking things on the computer.
And I guess one answer is that they're going to have to talk to each other. You're just going to need to have more different specialties working very closely early on throughout research projects.
Exactly.
Yeah. Joining us on Skype is an award-winning science fiction author, Kameron Hurley, author of, among other books, the God's War trilogy, where advanced bioscience plays an important role and for which she has won numerous awards. Welcome to Crosstalks.
Hello, thank you for having me.
Kameron, you've written about a far future world with very advanced biotechnology. How has today's bioscience research influenced you or inspired you?
Well, my novels really deal with biotechnological advances so far in advance of the research that inspired it, that in the societies I have, they're kind of regarded as magic. I loved what was said early on in the program about there's so many mysteries to be explored in this particular realm, and that's certainly very true of all the books that I write. It's that era of mystery that I love to look at. I actually took the inspiration for the technology that powers pretty much the entire world of my novel God's War and the sequels, Infill and Rapture, from some research done into both nanotechnology and the use of pheromones to manipulate insect behavior. So the idea is, okay, we can train wasps to sniff out explosives and we can use pheromones to change the behavior of bees. So why couldn't we then direct them to repair injury and cure diseases? And so I look at not only how we can use these sort of advances and technologies to change ourselves, but then also our environments. In my latest book, Mere Empire, I actually looked into research on plants and fungi and medical and architectural uses for these plant-like structures. So just as we've created, say, modern-day penicillin, I've explored other ways we could use compounds synthesized by fungi to cure infections, especially those which mutate very quickly, or use viruses to deliver specific changes to our DNA, like viral mediated gene delivery. So, which is already explored in labs, but I take all of these ideas and I push them to the next level. So I can have people who control and create these insects seemingly from nothing, and build the organic machinery that powers people and the world. And then also people who can do that with actual plants and form them into bridges and houses and other structures and save people's lives.
It's funny, the scientists are smiling and nodding, like this sounds completely reasonable, and I'm a little bit freaked out over here. Yeah. Kameron, what would you say are the most interesting or perhaps alarming things that you're finding out about biotechnology research when you're reading? Because clearly you're reading a lot.
Well, I'm very interested in our decreasing ability to fight infection, and both alarmed and interested in how we're going to combat viruses that mutate constantly with formulas that so far as I know, we haven't really changed much since we first kind of introduced them and said, okay, now this cures everything. And the problem is, well, the viruses are mutating and our response is not up to speed. So there's been some research done into the idea that we can harness antibiotics, which are actually created by human microbiota, which help regulate human health, which I think is a really cool idea. Now, as far as alarming things, about biotechnology, in my own research, I'm based in the US, and unfortunately, the bioscience industry, like many of our industries, is driven primarily by two things, and that is war and profit. So if the public good and the profit overlap, that's fantastic. It's great. But if a company does not see profit in investing in a cure, it's not quite as high up the list of things that they want to consider. So, I myself, I have an immune disorder. And as with many of these types of illnesses, companies have found there's more money to be had managing these illnesses instead of necessarily curing them. So that's something that I worry about. I find it a little alarming. Certainly that perhaps corporate greed may win out over the common good, as well as in other types of research. But I'm grimly optimistic. So we'll see what happens.
Good. Let's let the panel reflect on these two visions then. We were talking about the response to viral infections. Of course, I think everybody is if you believe in science, you're very concerned with the situation with antibiotics. And of course, right now, at what could be the beginning of a much even worse Ebola crisis, unfortunately, we are looking at these threats from these kinds of viruses. So the kind of knowledge that you are accumulating about the building blocks of the human body, will we be able to use that to solve these problems, or are you working on such a different level that we are not going to? That there isn't the practical application is so far away that it's something else. Mathias?
Absolutely, we will be able to use, and I think a very good analogy is what happened in the AIDS epidemic, which came in the 80s, and the people that got HIV infections, they were doomed, and they all basically died. The scientists put a lot of effort in the late 80s to try to find cures for HIV. Excuse me. And very quickly, there were some very good drugs being developed, and which now means that most AIDS patients today can live a normal life and even die at a normal age. Obviously, we now have the Ebola epidemic. It's kind of new. But I'm quite optimistic that the scientists will find cures for this, and we will see this. But it might take 5 to 10 years. But and hopefully then we will not have a very, very bad worldwide epidemic before that, like AIDS was. But there's another on the bacterial side. We have a problem because there is a misuse of antibiotics. And it does, and just like Kameron was saying, it means that the bugs starts to be resistant, and this is a problem. And here we have another problem, and that is that for the commercial companies, it's not so much money in developing new antibiotics. And this is a problem, and I think maybe society should go in and fund a little bit of that kind of research, because it's important for society, but it's not that commercially feasible.
When will you be able to hack a virus or hack infections? I mean, if you're on the building block level, can't you build us back together? Can't you put me back together when I'm broken? If you know what I meant to how my molecules are meant to function, if they malfunction, couldn't you reprogram me for the better?
Yes, I think that should be possible with using viruses again. Probably it should be possible to correct problems in the way cells have turned, and yeah.
But I mean, one of the problems, if I'm allowed to, is that we already today know several thousands of genetic diseases, And these diseases, you're born with a one slight change, and that means that you might be seriously sick or not so seriously sick. And we know very much in the lab how you can go in and correct that. The problem, though, is that in the body you need to do that correction in the right place.
Yeah.
And in all are trillions and trillions of cells. And you have to do it precisely. So it's this field that is called gene therapy, it's not that easy to do in practice. It's easier for blood diseases because you can take out blood and then you can do the change and then put it back. But if you have a problem in the lung, it's very much harder.
I think perhaps for a layperson, the problem here is that you have done so many miraculous things in the last decade or two that now it feels like science should be able to pull off every miracle. I can't compare the complexity of one miracle with another, so to speak. And even you are surprised at the results already. So that's also actually why it feels like turning to science fiction is so important, because somebody who looks forward at some visions of what we could be doing or what the consequences could be. I do have to ask you to reflect on profit as well. You said almost everybody who has AIDS today will live a normal life, or who has HIV today will live a normal life and not develop AIDS. And of course, that's not exactly true, because that is still a question of profit. It is a question of access to affordable drugs, for instance.
Yeah.
How do you view working for public institutions? How do you view the question of profit in your field, Erik?
So I think it's hard to say that. And of course, from one point of view, I agree with Mathias. It is a problem with the profit motives. But I also think occasionally, I think we might make it a little bit too easy for ourselves that that's a problem of the companies. Ultimately, it's a problem of society, because these companies are ultimately up to us as taxpayers. We could choose to fund this on the tax bill instead, which we normally don't. And that, of course, to some extent means that we are all greedy as taxpayers too, right? And large parts of the technology development that has made all this possible, that would never have happened without companies being involved. They're the difference to a large extent, that they have seen a profit motive here that has enabled all this to happen. So I think we are all a bit torn. The profit motive is important because that's what makes lots of these things happen. And you could argue that it's much better that people make money from curing ill people than from, say, building weapons or something. But then, of course, we all struggle with this. I don't think we have a patent answer to it. It's somewhat easier, in particular for me, that I think the closer you are to basic research, the less concrete this question gets. But unfortunately, I don't have the patent answer for you.
Looking back, a lot of notable works of science fiction in the past have quite accurately predicted innovations that would happen years later, sometimes 100 years later. But they've also missed a lot of things that today seem quite inevitable, and the invention of cell phones is the example that is always used of this. Cameron, what do you think that contemporary science fiction could be getting right about the future in regards to bioscience? And do you think that, and do you have an idea of where we will be wrong?
But, it's funny, as a science fiction writer, I get this question all the time, right? Predict the future. And I actually think it's not actually what we do. I would actually say that science fiction writers' place, like my place, is to inspire these technological changes. So we have these generations of researchers and scientists who grow up reading speculative fiction for these big ideas, for the possibilities that it shows them. And then they're inspired to go and make those advances real. So, I can't give you detailed schematics on how to power the world using bugs or create buildings from insect secretions or anything like that. But I write about the idea and its practical applications and get folks excited about those ideas, and I show them those possibilities. So stories are really the primary ways that, we've passed down information for tens of thousands of years. We're more likely to remember information or ideas passed down to us that way. So whether or not someone picks that up and runs with it is sort of up to chance. But largely what we're seeing, folks take the idea of the Star Trek communicator, and that really became the phone, right? And then it evolves into a smartphone. So I'm not going to make a prediction about what we'll get right or wrong. I'll say that I think it's my job to provide bigger and better ideas that inspire people to make those changes. And I want to show people,
Yeah, no, I just thought, I realized another thing the stories are very good at, of course, is simulating human emotional responses and sometimes thinking about societal change as well. And that's an aspect where I think science fiction has been incredibly valuable in predicting some of the mechanisms surrounding new technologies rather than the details of the technologies themselves. And of course, so a lot of research in the life science field today is focused on solving specific problems that affect us in contemporary life, like things like curing disease or diagnostic tools. It's not very far-fetched to assume that in the future we will have the ability to prevent and predict diseases, certainly even genetic diseases, perhaps deselect for certain qualities in humans, and so on. How do you think that this will affect us psychologically and politically? I'm going to ask Kameron first, and then I'm going to ask you whether this is even something you talk about. Okay, Kameron first.
Well, on the surface, looks like an easy question. Wouldn't it be wonderful? Cure all diseases. But on the other hand, I'd argue that we need to be very careful about what we classify as a disease. Some of those things I think we can agree on, cancer, autoimmune disorders, but what gets classified as needing fixing will vary by culture. I think that, is being hyperactive in the classroom a disease that needs fixing? Some of what's been categorized as a disease in the past has simply turned out to be normal human variations that individual societies, for whatever reason, rejected. So this becomes a problem when we look at issues of evolution and resistance to future diseases. So while we're mucking about with things that, maybe we don't totally understand right now, are we going to be deleting some of those things that we consider undesirable, but then in the process also taking out what might be a protection from a future disease or super plague, and we just don't know. So how we're affected psychologically, politically is really going to depend largely on how we choose to apply these advances, for good or ill.
Let's turn to the gentleman of the SciLifelab. As you're saying, the change is happening very fast. Do you have time to think about the world you're building?
I think we have, and I think it's also our duty to think about how our research can be applied. I completely agree with Kameron that some of these, we have to think about how this will affect the sort of society in general. I do think that viruses and bacteria we should fight. That's a war and we should win, but it will be a war. But other things I think maybe we should be a little bit more careful about. There is fantastic ways to reprogram cells and animals now, and I think that some of that is very, very good because we can produce nice products and energy and things like that. But other things I think we should keep away from.
What about you guys? Is this present in your work at all? Is this something that even at the coffee breaks?
I think it is,
Erik.
but I think it's difficult because many of these things, I would argue in particular in bioinformatics, which then again is a subject that historically has not come from the wet lab, but from, say, computer science, mathematics. So I think that you're suddenly having new generations or new fields of scientists that are suddenly being faced with very, very grave decisions. Could be about aborting a fetus or something that, and even though these scientists themselves might not necessarily they will obviously not make the medical calls. They are developing the tools that make it possible to make the medical calls. Ultimately, this is up to us as human beings. The decisions are very, very difficult, but I don't really think that ignorance can ever be an alternative to knowledge. But ultimately, we will have to make wise calls as humans. And I very much agree both with Mathias and the previous speakers here that we need to learn to accept that we can't simply start to erase every single weakness in human beings, because to tell the truth, I think most of us are a bit defect in our genes.
Matt?
Yeah, no, I agree. I think it's a really difficult concept. I think trying to purify our genomes and our genes, because it is, what is beautiful is very much in the eye of the beholder, and what is beautiful today might not be so beautiful in 100 years or in 50 generations. This we don't know. So,
is there an idea, though, this idea that we might accidentally deselect for qualities that might provide protection for threats that we're not yet aware of, for instance? Does this sound reasonable to you, or is there such a thing as an ideally operating body?
I don't think there is such an idea as an ideally operating body. We are constantly evolving, but I think we're quite prepared to what will come in the future. As we have a sort of a Gauss curve of individuals, and this is a wonderful way of how we are creating people by crossing with other partners to create this diversity. And if there is a catastrophe, I mean, that's what we learn from nature. There's always, well, not always, but at most of the time there are survivors, and then they will live on. I think just to comment a little bit about this, one of the things that we at SciLifelab I think is very important, and what we're betting on, is what some people call personalized medicine, and that is that we try to have a sort of a so good diagnostic so we know diseases before they happened. And this is a bet that we will take, that maybe in the not so far future, we will, maybe all of us take a blood sample every year, and we will then see diseases that maybe hasn't started to occur yet. And in some cases, such as cancer, this will be very, very good. Because if you see cancers early on, you can cure them very, very easily. If you find them very late, it might be too late.
And also some of our other big diseases, I'm thinking of degenerative diseases like Alzheimer's, for instance, which manifest in the brain very much, I mean, sometimes over a decade before you have symptoms. And we know how to treat it if we can find it. Yeah. And making those kinds of diagnostic tools widely available.
Yes.
Seems like a relatively small nut to crack in a way compared to some of the other things we're talking about here.
Yeah, although in the case of Alzheimer's, we don't have very good treatment. So it is maybe good to to see it early, because relatives will know what's going on, and that could be quite nice. Well, it's not nice, but it might be good to know it. But we don't have very good treatments. And that's very different from cancers, because if you find cancers early, you have very good ways to remove them. But obviously, it would be nice to have good treatments for Alzheimer's, find it early, and then cure it.
And we can do, but we can do more, and this is true with almost everything. The earlier we find them, we can do more.
But I think it's important to realize the stage we are at when it comes to many of these diseases, in particular in our brain, is that we're just trying to chart out the white spaces on the map. We haven't even begun to go to the areas and actually explore them, but we're realizing there might be a river here.
Yeah. This is very naturally leading me to my next question: is what can we not know? I mean, because sometimes when you speak to scientists, it feels like everything about human existence can be explained by biological phenomena. And I mean, and a lot of that has been like, well, nature or nurture, who knows? We're looking at a thing where perhaps even in our lifetime, we will have the answer to quite a lot of those questions. We will know what is nature and what is nurture. Do you have some kind of hazy vision of what the limits of life science knowledge is? What can we not know? What about humanity will not be a question of genes or cells ultimately?
Well, I would say that there's so much we don't know, and especially how the building blocks interact to create this fantastic organism like a human being. We're so full, and I think it, like Kameron was saying, it's so full of mysteries and magic, and there's so much that we don't understand, and we're just confused how our bodies can understand all the things that it's happening to and do the right thing all the time. And this is, of course, something that has happened over 1 billion years of evolution. But fortunately, we'd only understand a very small part of it.
Mats, what are you thinking?
Yeah, I think that would be very difficult to predict what would be the limit of what we can learn about us. And we will, as Mathias has been saying now a couple of times, we're so far away from that point that I don't need to concern about that in my lifetime. Okay.
Kameron, what do you see when we are going forward? What is the exciting limits of our knowledge now, or is that irrelevant to you because you can just make it up as you go along?
I was going to say, yeah, for me, I don't like to put limits on myself as a writer, because I think that if I'm going to be forward looking into the future, I need to be pushing the boundaries far more than anyone to inspire people, to push them a little bit farther than actual reality. So for me, yeah, there is absolutely no limit. I'm always at the margins.
Very good. I wonder now if we have any questions from the audience. I'm amused again to see that we have discussed pretty much everything about humanity in the course of a short conversation. Yes, in the back, please come to the podium and state your name and academic affiliation. Welcome.
Hello, my name is Annika Spetsches. I'm from KTH, and I would like to go back to the discussion earlier about neglected diseases. I think the word hasn't come up, but I think that was also what Kameron mentioned, also you mentioned. And in that aspect, I would like to ask the question, how much are you turning into a lobbyist, working with the World Health Organization, trying to promote your studies?
Interesting, yeah.
So I can actually say one thing there
Erik.
that I realized. I don't think that as a scientist, the most powerful role we have here is not necessarily as lobbyists, but in particular when it comes to the neglected diseases in the third world. Many of these biotechnology advances means that in a couple of years from now, you're going to be able to have diagnostic devices on cell phones, all these diagnoses that today cost tens of thousands of dollars, which means that we can only afford them in the Western world. They're going to be available for $2 in virtually the entire world. And ultimately, I think we're going to make a larger difference from that point of view rather than the lobbying.
Anybody else? Mathias?
I mean, I think all of us talks a lot to the, well, maybe not a lot, but we talk to the politicians, and this is both in Sweden, and I spend a lot of time in Brussels because that's where EU has its headquarters, and I also spend quite a lot of time in Washington, D.C., and obviously you are discussing with colleagues and politicians how you can use science to make a better world in the future. I wouldn't call it lobbyist because it's not that focused. It's more, we talk more general, I think most of us are scientists. But it's an interesting question because I think we also should involve ourselves in what should happen in the future. What should our tax money be spent on?
Well, yeah, because this is also where it gets important, is in relation to funding. You may not be thinking of yourself as a lobbyist when you are applying for money, but in a sense, you are affecting priorities in funding there with the solidity of your projects and so on.
Yeah.
And we do know from a lot of fields, I don't know about yours, that sometimes the direction of research is implicitly directed by the funding available. How much space do you feel you have to fight back when it comes, for instance, to neglected diseases?
I think there has been quite a renewed interest of, you call neglected diseases, but there is, of course, now in Europe and in the United States, there is something called orphan drugs, which are then, orphan diseases, which are diseases that affect very few people. And the society then, in both in the United States and Europe, has decided to put special rules around those diseases in order to promote research on these rare diseases. So there is a sort of a tendency to help out also not these neglected kind of diseases.
Good. Thank you. Thank you for your question. Do we have another question from the room? Yes, please.
So, hi, my name is Rutger. I'm also for KTH. My question is about ethics. So, how far do you go with your diagnosis of how healthy somebody is? For example, when the baby is still in the belly of the mother, if you can decide whether or not it's going to be healthy, how far do you go?
Yeah, I mean, I can answer that a little bit first. So obviously ethics is very important. We are now trying to start different initiatives where we look at wellness profiles in people. And it's very important, first of all, to get the approval for ethics, but also actually to decide what should go back to the actual individual about information about his health status. So we're not sort of screwing up people, but we're actually helping people to live a more healthy life. When it comes to your very difficult question about children, because it is a very, very difficult, and we have endless debates about exactly your question. What should the mother be able to know about the baby? And as you know, then we have had a relatively crude, I would say, way of looking at the fetus in the mother. But there are new technologies, thanks to what we are talking about here, where you only have to take a blood sample from the mother to know the genetic status of the children. And this is now starting to be implemented in several countries. And this, of course, creates the sort of problems that you are talking about. Should we allow this? Should this be allowed to even, and there's a very interesting one, and that is that it's very easy to determine the sex of the child. And should we allow abortions based on gender? And these are being treated in different countries in different ways. But I can assure you that everyone which is involved in this are constantly thinking about this, and we're trying to make wise decisions about how to apply these kind of diagnostics.
Mats?
Yeah. So, but still, I think so. We can develop the technologies to make better predictions, to enable more precise measurements of the state of fetuses. But I don't think it's really not. We shouldn't make the decisions on what the parent, what kind of decisions, or what. Well, we can help out, recommend what kind of information is useful for the parents. But still, it must be a political decision, I think, on how to deal with information in the end.
But even within the European Union, most countries right now are politically very polarized between value conservative and quite progressive, and sometimes the progressive is the same as scientifically or rationally oriented, and that is not always the case at all. It's different in different countries. But in the polarized political landscape, it's very difficult also to leave this to be a political question, because then perhaps what is correct or what is culturally acceptable could change at every election. So I think perhaps we're not going to have an answer to this question now, but I'm very happy that you raised it. Thank you.
Thank you.
Do we have time, I think, for one more question from the room? Yes, please.
Hi, my name is Anne. I'm studying Stockholm University. I was wondering how far bioinformatics can go with creating models, like organisms that used as models for testing medicine or modifying genes. I'm referring to those synthetics biology where they create bacteria, and you know what I mean? How far can bioinformatics go, or what is the chance of it?
Yeah,
that's a hard question.
Yeah, but it's exciting.
Had you asked me 10 years ago, I would never have guessed that we would have synthetic cells by now. So I think that's the first caveat. I think it's going to be very hard to guess how far we're going to be 10 years from now. The other problem is something we touched on before. I think you can get very, very far on the pure technical level. The problem here, as Mathias already alluded to, that the really complicated thing starts happening when you have an entire organism. A human body is more complex than just one cell. So I think we can do an amazing amount of things in synthetic biology, and even completely in the computers, predicting how, say, our new antibiotic should work and what molecules it will interact with. But at some point, we will still need clinical tests. We're going to need tests even on animals and at some point on humans before we start using this for real treatment. But of course, I think one such example is that we can likely remove 99% of animal tests in the future simply because we can do much, much more in computers and biotechnology.
Again, I will ask the question that scientists don't want to hear. When?
I would say 20, 30 years from now on, but that's likely going to mean that it happens in 5 or 10 years.
Great. Thank you for your questions. At the end of the show, we always like to ask our guests to look forward. And I'm sure we have grad students in the audience in the room or on the online in the online world who may not have chosen their specialties yet. So I will ask you all this, all 4 of you. If you could start out anywhere in the life sciences today and work on solving a specific challenge or mystery, where is the most important and exciting work going to happen next? And don't all say your own disciplines now. Like, what's the most exciting thing that's happening now?
The
Erik,
brain.
The brain.
Yes.
But anything in particular, or just figuring it out?
The entire brain is one big giant white field, right? That we're only starting to scratch the mere surface of it. There is an amazing amount of knowledge to be had there.
Last month on Crosstalks, we said that the ocean is the next frontier. Maybe the brain is a parallel next frontier to that. Yes,
that sounds
Mats?
good.
Yeah, I would start. I would probably take the other part of the body and try to address everything but the brain. Yeah.
Everything but the brain. Again, anything in particular that you want to solve?
Yeah, all the other diseases.
Everything else. Okay, very good. Matthias?
If I then change quite dramatically to sort of a medicine field, then I think it's interesting to think about reprogramming bacteria to create biofuels and bioenergy. And I think in 30 years, we might have new organisms that can take sunlight and carbon dioxide and create biofuels. Excuse me, in such a way that fuel might be, or energy might almost be for free. Huh. And that would, of course, change the whole planet. We can water the deserts and so on. So I think that's a very interesting perspective, that we can use life science to actually create almost free energy.
And maybe end war and starvation in the bargain.
Yeah,
that's also a pretty worthy goal. Now I'm thinking, why is anybody doing anything else except life science at this point? Yeah. Cameron, if you could start out anywhere in the life sciences today, where would you go?
Honestly, I have personal stakes in immunology, so I think that's very fascinating, the things that our bodies do to destroy ourselves, and I think that it could also be used to cure ourselves. So I think it's a very fascinating field.
Thank you. And thank you all. That's all we have time for today. I would like to thank our guests, Mathias Uhlén, Mats Nilsson, Erik Lindahl, and Kameron Hurley. And if you want to read Kameron Hurley, a great place to start is God's War. Crosstalks will be back on November 27th with two brand new topics. Until then, be safe and be brave.